Hi, this is Miss Nikki. Welcome to Chapter 8. Let's talk about the cell cycle. Hopefully we remember cell theory from the beginning of the semester. All cells come from pre-existing cells. This is called cellular reproduction. We're going to see cells grow. Also, this is what we need to develop into a newborn from a fertilized egg. So when they say fertilized egg, that's when you have egg plus sperm, and they come together, and then this is going to grow up into a baby, if I can spell. Baby. It's hard to do sometimes with this pen. So that's what they mean by fertilized egg, same thing as egg and sperm together, and we'll talk about this in, I think it's chapter 36 coming up in a few. So this cell cycle is showing you the stages that a cell has to go through in order to copy itself or undergo cellular reproduction. So the first is G1, which is growth, and S stands for synthesis. And G2, second growth phase, and the preparations for final division. So basically the cell is going to grow in size here. Then we're going to see the DNA copy itself. And then we're going to see organelles. Remember when we talk about organelles like mitochondria and endoplasmic reticulum and Golgi apparatus, all of those have to divide as well or copy in the preparation for division. Because what's going to happen, we're going to have one cell and it's going to copy itself into two cells. And depending on the chromosomes that you have in here, those chromosomes are going to copy as well. Chromosomes are the same thing as DNA, or we also saw the word chromatin, and all of that goes back to this S phase. Then we actually have this phase right here, which is called mitosis. So let me erase all of this up until now so we can get a good look at the slide. Okay, so let me try that again. All of this is what we call interphase. So interphase is made up of G1, S, and G2. Then we have this phase, which is called mitosis. Then within mitosis, we have phases as well. So think of test questions that could be asked. What are the three phases of interphase, or the three sub-stages of interphase? That's growth 1, or G1, S, and G2. Name the phases of mitosis. But remember that interphase plus mitosis plus something right here called cytokinesis, which we'll get to. Cytokinesis. All of that is equal to the cell cycle. So we need to know a little bit about some of these C words. There's lots of them. So chromosome versus a duplicated chromosome. Hopefully we can figure that out. It's just made a copy. So this is the same as this chromosome. It just copied itself, and now we have two. In the center here, what holds duplicated chromosomes together is something called a centromere. This will become important when we talk about mitosis in just a moment. So this is happening during S phase of the cell cycle. Duplicated chromosomes are also called sister chromatids. So if you have one chromosome and it copies itself, they're sister chromatids, and those are duplicated chromosomes. So mitosis. Some terms that you might see when we talk about mitosis, the first one is diploid or haploid. In mitosis, we're going to have a cell that's diploid, and it's an exact copy. It's going to make a cell that looks exactly like its original cell. We'll talk about haploid, but we won't talk about this until we get to meiosis. In meiosis, we're going to take a diploid cell and we're going to turn it into a haploid cell, and we'll talk about why once we get there. So here's a picture of the basics of mitosis, and then we'll go into each phase in just a second. So they went a little more simple here than a human, or the human number of chromosomes. In this one, they're only showing you 4 chromosomes. And this 2N means diploid, and diploid just means you have 2 of every chromosome. So let's say this is chromosome 1, and this is chromosome 1, and this is chromosome 2, and this is chromosome 2. So you have 2 1s, and you have 2 2s. That's what diploid means. So the basic here, you can see, we start off with 4. What happens? The chromosomes copy themselves. So this chromosome copied itself, and this chromosome copied itself, and the same with the blue. So if you have 4 chromosomes and they all copy themselves, you now have 8 chromosomes. And then these 8 are going to split into 2 cells that have 4 and 4, and that's what we have here. So count the chromosomes: 1, 2, 3, 4, 1, 2, 3, 4, and these 2 are identical, but these are also identical to what we started with. These are called daughter chromosomes, sometimes called daughter cells at the end. In human, we have 46 chromosomes. So the 46, they're going to copy themselves, and you're going to have 92. Then those 92 are going to split into 46 and 46. So here's a more detailed look at mitosis and each individual phase, and you'll see throughout that they say early, and then they have prophase. So they have early prophase, and then they have regular prophase. And then we're going to have early metaphase, and then we're going to have metaphase, sometimes even late metaphase. What they want you to understand is that this is a fluid process. Cells are undergoing this division or this mitosis gradually, and if you look at cells under a microscope, you might catch them in early prophase, or you might catch them in the middle of prophase, or you might catch them as they're transitioning from one phase to another. What I would suggest that you do is review each of these paragraphs that are under each title. So these paragraphs here, you need to know this information and exactly what's happening. So a test question might say, in which phase is the nucleolus disappeared and the duplicated chromosomes are visible? You need to be able to tell me that that's prophase. So what has to happen? We have our nucleus, right? Our nucleus has to break down, and they call that the nuclear envelope. The chromosomes have to duplicate themselves. And sometimes people will refer to all of this as prophase. Again, they're just telling you early and middle. So you can see the chromosomes have copied themselves. And you can see that there is no more nuclear envelope. And if you notice, these clusters here start moving farther away from each other. They're eventually going to be at opposite poles, kind of like North Pole and South Pole, right? These clusters, we'll see words for them a little bit later, but we should have seen centrosome and centrioles when we talked about plant and animal cells. And I talked a little bit about microtubules and filaments that were going to help the cell divide when it needed to divide. That's what those items are. So what happens in metaphase? I kind of skipped over early metaphase, but do you see how the centrosomes are moving to the opposite poles and that the chromosomes are kind of coming more towards the middle? Well, what's going to happen in metaphase is they line up in the middle. Sometimes they say they line up at the equator. So they line up right down the middle, and what's going to happen is this makes it really easy for these chromosomes to split, and then for half to be pulled one direction and half to be pulled the other, and that occurs in anaphase. So sister chromatids part and become daughter chromosomes that are pulled toward the poles. Another item that they mention, they talk about kinetochores, and I don't see it highlighted here in this picture, but do you see these yellow little spots on the side? Basically what they think is here are duplicated sister chromatids. They think that on the centromere, remember this was a centromere, not to be confused with a centrosome, which is what this area is, so you've got to make sure you understand these C words. On the centromere is a hook, and it's a protein, and it's a kinetochore, and that kinetochore binds to this spindle and actually does the contracting or the pulling to pull the chromatids to the opposite side of the cell in anaphase. And then the last phase is Teyla Phase, and this is where the daughter cells are going to form. And you can see on the top they show you animal cells, and on the bottom they show you plant. So there are different processes for animal versus plant. In the plant, a cell wall is going to build in between the two, and in an animal cell there's a pinching that occurs, and that pinching is called a cleavage furrow. So they do develop differently when we get to the cytokinesis or the end of telophase. So here's that word cytokinesis, or we're dividing the cytoplasm between the two daughter cells. And here's a better picture if you want to see the cleavage furrow. Basically, it's a contractile ring that pinches from the inside, and then eventually we separate these two daughter cells. Here is a cell plate. You can see the wall, the cell wall, is being made in the middle. So to talk about cancer, first we need to talk about cell cycle checkpoints. So we're back to our phases. Here's interphase, here's mitosis, and then we have these stop signs. These stop signs are called checkpoints, and these checkpoints are supposed to check the progress of the cell cycle of the cell to make sure that the cell can progress properly and that there's no damage. So in this G1, basically, if the DNA is damaged or the cell cannot be repaired, we're going to have apoptosis, which is programmed cell death. There's no reason to do this entire division if the cell is damaged, right? If we have a cell that's damaged, we don't want to copy it and make another cell that's damaged. During the S phase, we have DNA replication. Remember, this is when we go from one chromosome to duplicated sister chromatids, right? Well, G2, this is also the final growth for the preparations for division, so when your organelles are copying. So basically, we have another cell cycle checkpoint here at G2. So did the DNA replicate properly? If it didn't, if something happened in this process, then we're going to go ahead and initiate apoptosis or cell death. And then within mitosis, there's an M checkpoint. Basically, did the spindles assemble properly? So remember the spindles that come out and they grab the chromosome. Well, what happens if the spindles don't bind to the chromosome properly? Well, that's not going to allow the chromosomes to be pulled to the opposite side during anaphase, so you're going to have some chromosomal issues. So there are 3 checkpoints, and these 3 checkpoints ensure that we're not wasting time and energy if we're going to copy this cell. It would really stink if you waste all this time and energy to copy a cell, and the cell that you copy is bad. Then you have 2 bad cells instead of having 1, right? So these cell cycle checkpoints are there to make sure that everything is progressing properly. Now, how does this tie in with cancer? Well, cancer cells have found a way to evade these cell cycle checkpoints. So they're able to continue to grow and grow continuously even though they are damaged. So think of a tumor cell. It just keeps growing and growing and getting larger and larger. Even though it is damaged or the DNA is damaged, it's still able to evade these cell cycle checkpoints. The last thing I should mention is this G0. This G0 is kind of this little loop. These are non-dividing cells. So non-dividing cells, think of heart cells or brain cells where they're not actively duplicating themselves. So they're in this G0 phase where they're non-dividing. Okay, I'll see you next lecture. Bye.